Hello everyone. In this class, we'll learn about sex differentiation. Differentiation between the sexes of sex organs occurs throughout embryological, fetal, and later life. In both males and females, sex organs consist of two structures, the internal genitalia and the external genitalia. In males, gonads are the testicles, and in females, they are the ovaries. These are the organs that produce gametes, that is, egg and sperm, the reproductive cells that will eventually meet to form the fertilized egg, that is, zygote. As the zygote divides, it first becomes the embryo, which means growing within, typically between 0 and 8 weeks. Then, from the 8th week until birth, it is considered the fetus, which means unborn offspring. The internal genitalia are all the accessory glands and ducts that connect the gonads to the outside environment. The external genitalia consist of all the external reproductive structures. The sex of an early embryo cannot be differentiated because of the reproductive structures do not differentiate until 7th week. Prior to this, the child is considered bipotential because it cannot be differentiated as a male or female. Internal genital differentiation. The internal genitalia consist of two accessory ducts, that is mesonephric ducts, that is Wolffian duct system, and paramesonephric ducts in females, that is called as Wolffian duct system. The mesonephric system is the precursor to the male genitalia, and the paramesonephric is to the female reproductive system. As the development proceeds, one of the pairs of duct develops while other regresses. This depends on the presence of or absence of the sex determining region of Y chromosome, also known as SRY gene. In the presence of functional SRY gene, the bipotential gonads develop into testes. Gonads are histologically distinguishable by 6 to 8 weeks of gestation. Subsequent development of one set and degeneration of the other depends on the presence or absence of two testicular hormones, that is testosterone and anti-Mullerian hormone, also called as Mullerian inhibiting substance. Disruption of typical development may result in the development of the both or neither duct system, which may produce morphologically intersex individuals. Males. The SRY gene, when transcribed and processed, produces SRY protein that binds to DNA and directs the development of the gonad into testis. Male development can only occur when the fetal testis secretes key hormones at a critical period in early gestation. Testes begin to secrete three hormones that influence male internal and external genitalia. They secrete anti-Mullerian hormone, that is AMH, testosterone, and dihydrotestosterone. Anti-Mullerian hormone causes the paramesonephric duct to regress. Testosterone converts mesonephric duct into male accessory structures, including the epididymis, vas deferens, and seminal vesicle. Testosterone will also control the descending of the testis from the abdomen into the scrotum. In females, without testosterone and anti-Mullerian hormone, the mesonephric ducts degenerate and disappear. The paramesonephric ducts, that is Mullerian ducts, develop into uterus, fallopian tubes, and upper vagina. There still remains a broad lack of information about genetic controls of female development, and much remains unknown about female embryonic process. So, sex determining region of the Y chromosome, the SRY gene produces testis determining factor, which causes the multiple proteins that cause gonad medulla to differentiate into testis. Thus, developed testis produce Leydig cells, which produce testosterone, which leads to development of the Wolffian duct system into accessory organs and development of the male external genitalia, via dihydrotestosterone. And Sertoli cells also produce anti-Mullerian hormone, which causes regression of the Mullerian duct. External genital differentiation, development of external genitalia. By 7 weeks, a fetus has a genital tubercle, urogenital groove, and sinus, and labioscrotal folds. In females without excess androgens, these become vulva, that is clitoris and labia. Males become externally distinct between 8 to 12 weeks as androgens enlarge the phallus and cause urogenital groove and sinus to fuse in the midline, producing an unambiguous penis with the phallic urethra and a thin rugate scrotum. Dihydrotestosterone will differentiate the remaining male characteristics of external genitalia. Sufficient amount of any androgen can cause external muscularization. Most potent is dihydrotestosterone generated from testosterone in skin and genital tissue by the action of 5-alpha reductase. In male fetus, may be incompletely masculinized if the enzyme is deficient. In some diseases and circumstances, the other androgens may be present in high enough concentration to cause partial or complete masculinization of the external genitalia of genetically female fetus. The testes begin to secrete 3 hormones that influence the male internal and external genitalia. They secrete anti-Mullerian hormone, testosterone, and dihydrotestosterone. Anti-Mullerian hormone causes paramesonephric ducts to regress. Testosterone, which is secreted and converts the mesonephric duct into male accessory structures such as epididymis, vas deferens, and seminal vesicles. Testosterone will also control the descending of the testes from the abdomen into the scrotum. Dihydrotestosterone, also known as DHT, will differentiate the remaining male characteristic of the external genitalia. Further sex differentiation of the external genitalia occurs at the puberty when androgen levels again become disparate. Male levels of testosterone directly induce growth of the penis and indirectly the prostate. Thank you.